Hello friends, welcome to Bosons Botany. I am starting with chemical composition of RNA, basically mRNA, rRNA, tRNA. For all these three types of RNA, the chemical composition will be the same. So, the structural compositions are nucleotides or ribotides for RNA. This is the structural composition. The ribotide or nucleotide is the structural composition of an RNA. Each of these are linked together via a phosphate group to form a strand of RNA known as polyribonucleotide chain. Similarly, if you recall the structure or the chemical composition of DNA, with the help of phosphate groups forming phosphodiester linkages, we form polydeoxyribonucleotide chain. Similarly, for RNA also, using a phosphate group, these phosphate group continuously form a phosphodiester linkage or several phosphodiester linkages to form a polyribonucleotide chain, which is known as RNA. Now, these are the components which we require to make a polyribonucleotide chain. The first one is nitrogenous base or nitrogenous bases. We have purine and pyrimidine. Under purine we have adenine and guanine having one 6 carbon chain, cyclic chain, and 1 5 carbon cyclic chain. This is purine, adenine and guanine. Under pyrimidine we have cytosine and uracil in place of thiamine. Thiamine is found in DNA, but uracil is found in RNA only. And the structure for cytosine and uracil is a single 6 carbon cyclic chain. The second component we need to prepare RNA is ribose pentose sugar. Same for DNA also, but at carbon number 2, if you count the carbons here, their positions, carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5, 5 carbon compound, but at carbon number 2, the hydroxyl group is intact. The oxygen is not replaced, it is there in place, and therefore RNA is labile, or it is not inert under room temperature, or it is very reactive as compared to DNA due to the presence of this hydroxyl group. And why it is labile, why it is not stable as compared to DNA? Because there is electrostatic repulsion or electrostatic force of repulsion between these two hydroxyl groups, because these two are very bulky, very huge molecules. They repel each other, and due to that the whole structure is very unstable, and that is why RNA is not stable compound or structure as compared to DNA, because in DNA at carbon number 2 the oxygen is removed and we only have a hydrogen left here, so no chance of electrostatic force of repulsion. But in RNA there is a force of repulsion experienced, therefore RNA is not very much stable and it is a labile structure. Now, sequentially, if you recall the nucleosides and then nucleotides, the nucleosides, but in case of RNA we will say nucleosides as ribosides. The ribosides are compounds which are composed of nitrogenous bases and sugars. So when nitrogenous bases and sugar are bonded together or linked together, we get ribosides. So there are 4 main types of ribosides. First is ribose pentose adenosine. At carbon number 1 there is a adenine group linked, and therefore it is ribose pentose adenosine. Similarly, if you link a uracil here at carbon number 1, we will get ribose pentose uridine. So just remember the terminology I am using here. uridine, not uracil. Many a times people make mistakes by writing uracil, so it is uridine. Similarly, ribose, pentose, cytidine. If you link a cytosine nitrogenous base at carbon number 1, then the terminology will become ribose, pentose, cytidine, not cytosine but cytidine. And for guanosine, it is ribose, pentose, guanosine. I am not I have not drawn the structures here because it is just exact of these two structures that I have drawn. Only you need to replace adenine by cytosine here and guanine to make ribose, pentose, guanosine, but the structures are similar. Now if you link a phosphate group at carbon number 3, then we will form a ribotide or nucleotide. So ribotide is formed by the linkage between riboside and a phosphate group. So same, if you take the example of this, only one single example, and remaining examples will remain the same. Only you need to remember and understand the concept of riboside and ribotides. So if you take this ribose, pentose, adenosine, and if you link a phosphate group at carbon number 3, then you will get ribose, pentose, adenosine monophosphate. Just add this word here, monophosphate. That's it. Similarly, if you take this ribose, pentose, uridine and attach a phosphate group at carbon number 3 like this, then you will get ribose, pentose, uridine monophosphate. And this is the phosphodiester linkage, and during its formation a water molecule is removed. This is a condensation reaction, and during a condensation reaction byproduct is always removed, and in this case a water molecule is removed. So this is also same for ribose, pentose, cytidine monophosphate and ribose, pentose, guanosine monophosphate. So this is all about RNA. Just in this way you can form a single stranded chain of RNA. Just repeat this structure for uridine, for cytidine, guanosine, and you will get a polyribonucleotide chain by forming phosphodiester linkages attaching phosphate groups at carbon number 3. So this is all about chemical composition of RNA. In my next video, I will take up the structures and some important features about mRNA, rRNA and tRNA. So please watch that video also, and hope you understand this topic very clearly. And if you have understood, please hit the like button, and also hit the subscribe button down below, and press the bell icon to get fresh notifications from Bosons Botany. And if you have any doubt, anything you want to share, please write down on the comment box below. And thanks for watching my video. Thank you.